Hey guys, Miss Cote here, and welcome to the last episode of the genetic stuff and our heredity unit. In an earlier video, we looked at how two characters found on two different chromosomes are inherited independently of each other. But what if those two characters are found on the same chromosome? In that case, they are most likely inherited together. In this video, we're going to look at this situation of genes being found on the same chromosome. This is referred to as linked genes. And then we'll be finished with chapter 15. So let's get started. Along with the experiments Morgan did on sex-linked inheritance, he performed another experiment important to our understanding of linked genes. He looked at two genes, one for body color and one for wing size. First, he started with the parents. One parent was homozygous for the wild type for both body color and wing size, which in this case is gray and normal. The other parent was a double mutant. This fly had a black body and vestigial wings. Notice the symbols being used represent the mutant phenotypes, V for black and VG for vestigial. As expected, all of the F1 generation were wild type for both genes, but heterozygous for both. Now, instead of mating two of the F1 flies together to get the F2 generation, like we've been seeing in these problems, Morgan did a test cross. He wanted to double check his work and take advantage of the heterozygous genotype in the F1 offspring. He mated an F1 female with a male who was a double mutant, so black body and vestigial wings. So if you did a Punnett square of this cross, you could potentially get four different phenotypes. Wild type, which is gray body and normal wings, black body and vestigial wings, gray body and vestigial wings, or black body and normal wings. Morgan predicted that if the genes were on different chromosomes and assorted independently, he should see a 1 to 1 to 1 to 1 ratio. You would see all four phenotypes. If the genes were on the same chromosome and therefore inherited together, he should see a 1 to 1 to 0 ratio. All offspring would look like the parents. The results of his experiment were somewhere in between. While there were a large number of offspring that looked like one parent or the other, we call these parental phenotypes, there was a small number that were a mixture of the two parents. We call these recombinants because they are a mixture or have a combination of traits from both parents. Due to the large number of offspring that showed the parental phenotype, Morgan suggested that these genes for body color and wing size were on the same chromosome. But how could he explain the recombinant phenotypes? He proposed that if the genes were on the same chromosome, in order to get a mixture of traits from each parent, the physical connection between the two genes must be broken somehow. Turns out there is such a process that can break off a piece of a chromatid and attach it to a non-sister chromatid. And remember, that process is crossing over, which happens during prophase 1 of meiosis. That would explain the small amount of recombinants. So if we add to the original model and put in the chromosomes and genes, we can see how this can come about. When the F1 generation makes its gametes, we see that crossing over can take place during meiosis 1 in the females. In case anyone is wondering why it doesn't show crossing over in the males, it's simply because even if it did happen, it wouldn't change anything since both chromosomes are the same. And the model clearly shows how we get the possibilities in the offspring. Another thing you can do with this is to calculate how often recombination can occur. Because, College Board loves to throw math stuff at you whenever possible. This is referred to as recombination frequency. And this is pretty simple. All you need to do is calculate the percentage of recombinants. First, you add up the number of recombinants. In this example, it's 206 plus 185 equals 391. You divide it by the total number of offspring. So we have 985 plus 944 plus 206 plus 185, and that equals 2,300. And you multiply that number by 100 to get a percentage, which in this case is 17%. The part divided by the whole times 100. Keep in mind that a recombination frequency less than 50% usually means the two genes are linked. If the recombination frequency is 50% or more, then that means one of two things. The two genes may be physically linked, meaning they're on the same chromosome, but far enough apart to behave as if they are unlinked. Or they could totally be on two different chromosomes and definitely unlinked. Obviously, creating these recombinant offspring leads to more genetic variation in the population. But being able to calculate recombination frequencies can also help us figure out their relative positions to other genes on a chromosome, something called gene mapping. One of Morgan's students, a guy called Sturtevant, and no offense to him, but you don't need to memorize his name, used recombination frequencies calculated from their experiments to create a genetic map. His reasoning was simple. The higher the percentage of recombination, the further apart on the chromosome the two genes were, and the higher chance that crossing over can happen. If the recombination frequency is low, then the genes are relatively close together and you shouldn't see a lot of crossing over happen. In other words, the probability of a crossover between two genes is proportional to the distance separating the two genes. This diagram shows how a linkage map can be created based on recombination frequencies. You can take the number you calculated, remove the percent sign, and add the unit map units to it. Now, this linkage map does not tell you the precise locations of these genes on a chromosome. There are other ways to figure that out, but linkage maps can tell you the relative distance and order of one gene to another gene. Here we see that the recombination frequency of 17% between black body color and vestigial wing size is expressed as they are on the same chromosome and are 17 map units away from each other. Additional experiments in calculating recombination frequencies shows that the gene for cinnabar eye color falls approximately between the two original genes we looked at. I know the numbers don't align perfectly, but they're close enough for our estimations. Even more experiments produced more data to produce a linkage map involving several genes. What College Board wants you to be able to do is recognize when genes are linked, be able to calculate a recombination frequency, and to be able to construct a linkage map using multiple recombination frequencies. And this is something we will practice in class, so please pay attention when we do it. Since this chapter is all about chromosomal inheritance, as opposed to just the inheritance of genes, any abnormalities of the chromosome itself can affect the gene being expressed and therefore the phenotype. Two things that can happen are that the offspring can end up with the wrong chromosome number, or there could be some type of change in the structure of a chromosome. So let's take a look. Having the wrong number of chromosomes stems from when the chromosomes separate during meiosis. This failure to separate is called non-disjunction and could happen either during meiosis 1 or meiosis 2. The end result is that instead of ending up with half the number of chromosomes in the gamete, there may be an extra chromosome or the gamete could be missing a chromosome. Over on the left demonstrates non-disjunction during meiosis 1. The homologous chromosomes do not separate during the first division. So at the end, it affects all of the possible gametes. 2 have an extra chromosome, while the other 2 are missing a chromosome. When non-disjunction occurs during meiosis 2, when the sister chromatids do not separate correctly, only two of the possible 4 gametes are affected. One will have an extra chromosome, while one will be missing a chromosome. The other 2 gametes see no change, having the correct number of chromosomes. Remember, which sperm and which egg come together at fertilization is completely random. So if one of these gametes that experienced non-disjunction is involved in fertilization, the final chromosome number of the organism will be changed. This is called aneuploidy. Ploidy refers to a set of chromosomes. We've seen that haploid equals 1 set, while diploid equals 2 sets. And the prefix anu refers to not good or not well. So not a good set of chromosomes, meaning something is wrong. Offspring with this condition have an abnormal number of a particular chromosome. For example, humans have 46 chromosomes. A monosomic zygote would have 45 chromosomes, meaning it would be missing one, while a trisomic zygote would have 47 chromosomes. It would have an extra one. In some cases, total non-disjunction may happen, and the new individual inherits not just an extra chromosome, but a whole set of chromosomes. This is a condition called polyploidy and is more common in plants than in animals. I don't know if you've ever extracted DNA from strawberries in a previous biology class. When you pull the DNA out of the test tube, it looks like a big glob of snot. But the reason why strawberries produce a good amount of DNA is that they are octoploidy. They have 8 sets of chromosomes. There are 4 ways the structure of a chromosome can be altered. This slide gives you a description of all 4, and you can pause this slide if you would like to write down the definitions, but I want to show you by using a diagram. So off to the next slide. In deletion, you're actually removing a part of the chromosome, so any genes located in the deleted part no longer exist. The opposite happens during duplication. A piece of the chromosome is repeated, so you can end up with multiple copies of a gene. In inversion, a piece of the chromosome is flipped around. You're not adding or deleting any DNA, but you are changing the order in which genes are found on the chromosome. And finally, translocation is when a piece of a chromosome breaks off and attaches to another chromosome. I don't have time to go over the rest of this section. You guys can read about different human examples of these chromosomal alterations, some which you may already know about. And remember, it's always good to be familiar with examples. The last part of this chapter talks about some exceptions, because there are always exceptions. But I do want to mention them because you never know when these might pop up. The first exception is called genomic imprinting. This happens only in mammals and only for certain genes. Basically, for these genes, the expression or non-expression of a gene depends on which parent passes on the gene. In this example, the expression of the IGF2 gene depends on which allele was contributed by dad. Whatever allele mom passes on doesn't count. So if dad has the normal allele, the offspring mouse will be normal sized. But if dad has the mutant allele, the offspring mouse will be a dwarf. Notice that mom has the normal allele, but mom's allele is silenced and therefore not expressed. Pretty weird, I know. The other exception involves extranuclear genes. If you remember, not all DNA is found in the nucleus. Organelles such as mitochondria and chloroplasts contain their own DNA. And all organelles are inherited from mom. In reproduction, the sperm only contribute their portion of the chromosomes for the new individual, which is why the egg is much bigger than the sperm. The egg not only contributes their portion of the chromosomes, but all the cytoplasm and organelles as well. Why this is important in mitochondrial genes are obviously involved with cellular respiration, providing energy for the cell. So if mom has a defect or a mutation in her mitochondrial genes, usually leading to some sort of metabolic issue, then all of her children will inherit the same disorder. I'm also going to throw in the fact that there are new studies showing that dad can pass on mitochondrial DNA, but I'm not going to go into that because I just want to keep it simple, and we really don't know what's going on with that yet. Wow, so that was a lot of information in one video. Thanks for hanging in there. This is going to wrap up Chapter 15 and our heredity unit. So until I see you next, be a good human.